Hello, I'm delighted to be here and will be presenting on several types of multimodal data fusion approaches. What I will start with is just a brief introduction, just to kind of introduce multimodal fusion. Obviously, we have many different data sets that we are collecting these days. The goal of fusion is to identify the joint information that might be otherwise invisible to one or more modalities. You can think of multimodal fusion along a spectrum where on the left we have visual inspection, where we have two different data sets analyzed separately and we sort of visually combine them. Data integration is something like PET-CT fusion, where you co-register modalities. Asymmetric fusion is constraining one modality with the other, so EEG constrained by fMRI activity, for example. And symmetric fusion is jointly analyzing multiple modalities at once. So we have this spectrum from low to high information. You can also think of late and early fusion, where late would be, I've computed a bunch of different features, maybe regions of interest or volumes, et cetera. You can do thousands of those, analyze them all at once. So that's kind of a late fusion, whereas early fusion allows the modalities to interact earlier, and that's more like the symmetric fusion I mentioned. Finally, we can look at multimodal fusion in a univariate way, where we look at individual voxels and we pair those voxels up with other voxels in other modalities or time points, etc. We could also look at multivariate, in which case we're looking at patterns of activity across modalities that are linked. So there's a lot of fusion approaches out there these days. We talked about symmetric, asymmetric, model-driven, and data-driven approaches. And here we have different kinds of blind source separation, and then you can have semi-blind where you have some constraints. So there's a lot of choices out here, and it's hard to compare these approaches to one another. Data fusion approaches are increasing, and it has been shown pretty consistently that it tends to be helpful both in increasing accuracy as well as revealing information that you can't see separately, right? So white matter patterns linked to functional patterns, for example. So I think there's a real need for a unified framework to try to facilitate comparison and implementation of models in the context of these large and more complex studies. So we can look across two different spectra, single and multiple data set. So here we have one data set, here we have maybe multiple modalities, multiple time points, et cetera. Unidimensional and multidimensional sources. So unidimensional would be every source is separately estimated. So this would be like independent component analysis. Or we can impose subspaces where maybe we have groups of sources with higher dependence, and then there's independence among subspaces. So it's in this context that we presented this multi-data set independent subspace analysis unifying framework. So this is work done by Rogers Silva at Trends. And essentially we're optimizing a common cost function, KL divergence, Kullback-Leibler divergence among all possible mixing combinations, subspace structures, and for all types of data, input data. So I'm not going to really go into the details of the method. It's in the papers below, but it really is a unifying toolkit. We show that it provides good performance even with low SNR. It's a very general formulation, and it allows us to optimize using a nonlinear constraint optimization. In the context of MISA, you can implement, for example, multi-data set, multi-dimensional sources. And this allows us then to implement any of the other models, single data set and unidimensional as well. So we can implement all of these models below and more, including ICA, joint ICA, canonical correlation analysis, linked ICA, partial least squares, etc. One of the ways that that we've been looking at this is characterizing the algorithms along what's called the multi-diversity triad. So diversity in this context is a property, a constraint, or an assumption that gives us a unique solution. And so we can think about multi-data set diversity, where we have multi-data sets or multi-views. One observation, for example, is that the more data sets or views we have in an M-order tensor decomposition allows us to estimate more unique sources. This assumes a hard linkage. Essentially, we're concatenating data sets in multiple dimensions. Statistical diversity would be, are we optimizing for second order statistics or higher order statistics? It allows for soft linkage among modalities and concepts like, do we want to enforce sample dependence or assume stationarity or non-stationarity? Finally, constraint diversity would be, are we enforcing sparsity or smoothness or similarity to a prior, like maybe diagnosis or a cognitive score used to guide the solution. So let me just give you 3 quick examples of blind source separation fusion models. The first is joint ICA. In this case, we have 2 or more modalities. We're assuming a common mixing matrix A, multiple modalities, and we're estimating modality specific sources mixed with this common covariant mixing matrix. So this assumes multiple data sets, unidimensional, maximizes independence. Linked ICA is a similar model with the added flexibility of this source-specific scaling matrix D. This assumes a multi-data set, unidimensional, and maximizes for independence as well. And then finally, the most general framework is the MESA framework. And this again assumes multiple modalities. It allows for multiple mixing matrices and multiple modality-specific sources. The sources can also have an arbitrary subspace structure imposed on them or estimated from them. So for example, in this case, we have 2 sources within this red subspace in this modality, and that subspace also spans to a different modality here. So next, I'm going to just show an initial analysis from some real data where we're implementing what's called the transposed independent vector analysis model within the MESA framework. So in the transposed IVA model, we have, say, voxels in this direction and subjects in this direction, and we're essentially identifying a common profile across data sets. We're using 3 modalities: gray matter segmentation maps, amplitude of low frequency fluctuation from fMRI, and diffusion fractional anisotropy maps. We'll be looking at 2 data sets, 3,000 subject data set from the UK Biobank and 1,000 subject patient data set, including individuals along this psychosis spectrum. So just again, one slide to show you the kinds of output that we obtain. When we're working with unidimensional sources, we get these triplets here where we have structure, function, and diffusion. And you can see here relationships that were identified along cognitive variables, fluid intelligence, prospective memory task. Age is a large driver of variance in the UK Biobank data. You can see it here from young to old, and then sex-associated variation here. In the patient data set, if we just look at the bottom here, one of the components was identified showing both age-associated and patient variation in a consistent manner across all modalities. We identified these patterns that were previously identified in unimodal analysis. For example, this bilateral insula temporal and medial frontal region in schizophrenia patients. Here you can see the age relationship and the patient relationship. All patients are showing on average reduced loading on all 3 modalities. So now let me transition to another type of subspace, which is the complexity of the result. So what we can do is, if we look, for example, just at functional data for the moment, and we think about domains, functional domains as modalities. So these are ICA estimated domains. And we allow the model to evaluate the complexity that's required to maximize accuracy in a classification task. So in this case, schizophrenia versus healthy controls. And what we found was, high complexity between domains like sensory motor and cognitive control, low complexity within domains, and lower complexity, for example, between cerebellum and subcortical areas. So this gives us some, and this was also, I should mention, replicated in an independent data set. And we found very similar complexity results when we did that. We can extend this to a full multimodal pipeline. Here we'll just look at functional MRI, and structural MRI. And so there's a number of steps to this pipeline, which I'm going to sort of gloss over, but there is a paper here you can refer to. And then it gives you this kind of result where we can look, for example, at structural complexity and functional complexity. They were, somewhat consistent, where we found high complexity in auditory, visual, default mode network in both modalities, lower complexity in subcortical to cerebellar, suggesting that those are potentially communicating more with these other domains, and it thus requires a less complex model to pick up, perhaps a nonlinear, just a linear model in some cases. Finally, dealing with multiple scales, right? So oftentimes when you try to combine structural MRI with functional MRI, we squash fMRI down to a network or to an ALF map or something like that. And in that case, we've lost a lot of information. We're fusing it later in the process. So to avoid that, we developed an approach called parallel group ICA plus ICA, where we're doing a group ICA on the fMRI data and an ICA on the structural data. So that's called source-based morphometry. It's like a multivariate VBM. We're identifying components that have common covariation among subjects. And in order to link the two, what we do is we take the individual subject networks or maps that come out, and we evaluate the similarity between that subject and its mean, the mean across all subjects, which provides a subject profile vector for each component. We then link those between modalities in the algorithm. So now we have this algorithm that we're optimizing across those different metrics. This gives us a lot of advantages. It actually we show that we can have enhanced sensitivity to identify links that are present between the modalities compared to doing it separately and post hoc trying to link them. We also obtain additional information. So we have the maps, but we also have functional network connectivity, which we can extract from the model after it's fit and look at the relationship between components, for example. We've extended this to three-way group ICA fusion. So in this case, we have fMRI, which is a temporal modality, and then we have two spatial modalities, structural MRI and diffusion MRI. Again, looking at simulations, this approach gives us enhanced sensitivity to identify links that exists, and it doesn't inflate and produce links that are not there. Looking at one result from schizophrenia data set, we see a component here that has some common regions we see from fMRI, cerebellum, sensory motor, and subcortical, as well as a gray matter pattern, again, somewhat similar to what we find typically, and fractional anisotropy here. Importantly, when we took the initial results and then we fixed the regions and we evaluated it in an independent data set, those links were replicated. And so in sum, multimodal analysis, it often provides benefits over unimodal by enhancing accuracy, allowing us to evaluate joint patterns, and it can capture otherwise invisible joint information. Flexible and unifying approaches like MESA, I believe, are key to meet the increased complexity of multi-data set tasks that we have, especially in the context of large N population studies. It allows us to impose subspaces or estimate subspaces, and allows us to do performance comparison across different models, because we have now a common implementation, a common cost function. Subspace model complexity can help us improve performance and also provide data-driven insights into the problems being studied by evaluating the model that was fit to the data. And finally, we can handle, if we handle mismatched dimensionality and move the fusion earlier in the process, we can minimize information loss and improve our ability to link across modalities. With that, I'll just mention that we try to implement all of our approaches in tools, software tools for the community to use. So we have our GIFT software, the Fusion ICA toolbox, which will soon be including MESA. You can also get the code here from Roger's GitHub site. And I want to thank everyone at the Trends Center who has contributed to this work, as well as our many, many collaborators and our funding sources. With that, I will close. Thank you very much.
It's my pleasure to talk to you about our new method for identification of scanner effects for denoising imaging confounds from large-scale neuroimaging data sets. Small sample sizes in neuroimaging studies are really problematic because of their poor sensitivity or low power to detect true effects, which in turn leads to a high rate of missed effects. Low sample sizes also lead to biased estimation and inflation of significant effects, which was shown very nicely in the study from 2017 using HCP 500 social cognition task fMRI data. The top panel shows a spatial map of correlations between brain activation in response to social stimuli and a measure of personality trait agreeableness. The peak value at any voxel in the brain was 0.25 for this correlation. Random subsamples with 30 participants in each subsample and reanalysis to estimate the association between brain activation and agreeableness showed that the peak voxel, the voxel with the peak correlation value varied across all the subsamples, and that the actual correlation strength was highly inflated, being more than double what the peak correlation was in the full sample size. Small sample sizes are also very problematic for machine learning algorithms, leading to overfitting and inflation of accuracies of prediction accuracies, and large errors for prediction and classification. Jolens showed in 2019 using simulated and real imaging data with both small effect sizes and large effect sizes. In this case, the imaging data, the outcome was a measure of intelligence for small effect size, and for large effect size, the outcome measure was age. They showed that sample sizes of 400 plus are really necessary to achieve a decent out-of-sample performance for these different regression machines, including multiple regression and elastic nets. Veracqua also showed that small sample sizes lead to inflation of prediction accuracy. So as the sample size increase, the prediction accuracy drops off. And this is also in part due to the fact that when you increase the sample size by combining data across different data sets or studies or sites, you introduce more sources of biological variation and non-biological variation. The biggest source of non-biological variability, which is something that we care absolutely nothing about, are site and scanner-related variability. And these sources of variation represent a major barrier to pooling MRI data across studies or sites. This variation is due not only to fundamental differences between different scanners, but also to differences in acquisition parameters that may be used across the different sites or studies, and hardware and software upgrades that may occur within a scanner. These effects are compounded in multi-site longitudinal studies in which you have between scanner variability, so due to different scanners, and within scanner variability that may be related to hardware and software upgrades. These effects have been shown to impact all MRI modalities and can confound the detection of true effects of interest and complicate the interpretation of results. So what can we do about this if we want to create larger neuroimaging data sets by pooling data across sites or studies? Well, there aren't too many methods for obviating scanner and site effects. You can harmonize the data collection and or processing across studies and sites, but ultimately you will always still have differences due to the fact that the scanners are different, and no degree of harmonization will completely eradicate fundamental scanner differences. So it's also common to try to remove these effects or control for them post hoc using some kind of regression technique. In the simplest case, you construct a model as a simple regressor or set of regressors of values that encode site or scanner or study. The regressors are included in the group level statistical models to control for the effects, or they're used to denoise the data prior to statistical modeling. However, these kind of regressors may be a poor proxy for these effects. They certainly don't take into account any kind of within scanner or within site variability that can occur during the data collection period. And these effects are more problematic the bigger the data set. So with much larger sample sizes, there's not only more sensitivity to real effects of interest, but unfortunately, there's also a greater sensitivity to confounding effects. So we really have to be careful with this issue when we're trying to combine data to create really large neuroimaging data sets for our analyses. Data-driven methods can help because they can provide a better model of these kind of effects. And people are really familiar with ICA-based denoising technique, which is commonly applied at the single subject level for denoising motion, physiological, and scanner session artifacts from fMRI data using some automated tools or even doing it by hand. This approach has also been applied to multi-site data, in which ICA of an individual modality was used to construct a data-driven model of the noise effects, and regression of the noise patterns out of the original data removed the effects. Noise components are identified by correlating the subject loadings, because in this case you have a spatial map for each subject, you concatenate it across all subjects, and then you do ICA, and you get spatial patterns and subject loadings on each pattern. The noise components are identified by correlating those loadings with site or scanner variables. So it seems a bit circular, why not just use the site and scanner variables in the denoising procedure? And the answer is that the subject loadings actually will capture some of the within site or within scanner variability too, not just the between site or between scanner variability. There are challenges with ICA denoising for multi-site, multi-study MRI data. Namely, you have to apply it to each modality separately. So you might have task fMRI data, you may have diffusion MRI outcomes like fractional anisotropy and mean diffusivity. You may have structural MRI outcomes like gray matter concentration, peel surface area, or cortical thickness. You would have to apply ICA to each one of those sets of spatial maps separately. And there may be hundreds of components for each of those modalities that you have to wrangle, which, that requires implementing ICA to achieve a good separation between signal and noise, and labeling noise and signal components, both of which are kind of hard to do, and there's no good automated methods that exist. It may be difficult to develop automated methods too, because the scanner side effects are likely dataset specific, depending on the particular combination of scanners that were used to collect the data. So we need new methods for tackling this problem. The approach that we came up with proceeds as follows. You do QA and pre-processing of the individual modalities for all the participants, and then apply data fusion to all modality subject series simultaneously. Identify multimodal scanner-related components from the decomposition, and then denoise the scanner effects via regression to produce clean data with reduced scanner effects that can then be used for further higher-level modeling, refined data fusion, classification, machine learning algorithms, etc. So, this method was really brought to life by Dr. Huanji Li, who was a postdoc at the time, and is now faculty in Dalian, China. So the data fusion approach we use is linked ICA, and this cartoon from Groves in 2011 shows that you process each MRI data type, so diffusion and structural MRI shown here, through the standard processing pipelines to identify FA, MD, and MO tensor mode maps in this case, gray matter concentration, peel surface area, and cortical thickness, for example, for each participant. For each modality, then, the data are concatenated across subjects, and all modalities are fed into the linked ICA simultaneously. The linked ICA will decompose this multimodal data into a set of multimodal spatial patterns and associated subject loadings. And you just obtain one set of subject loadings per spatial pattern, and the subject loadings reflect the strength of that pattern in each subject. So this pattern is common to all the subjects, but the subject loadings reflect the between subject variability in the strength of the pattern. So what really inspired us to develop this approach was the next paper published by Adrian Groves in 2012, in which he used linked ICA to look at aging effects across the lifespan. But he also reported several components that were associated with the scanner software upgrades that occurred during the data collection process. And so when I saw this, in this case, we have a multimodal diffusion pattern whose loadings, subject loadings, were associated with scanner software version. I thought, maybe we can harness this technique to do data denoising of scanner effects in a kind of principled way that might be a little bit easier and more robust than the approaches that were available at the time. So to demonstrate our technique, we used data from 156 participants from 6 different studies of chronic cannabis use that were conducted at McLean Hospital on the same Siemens 3T scanner, but spanning different software versions, 3 different software versions, as well as a massive hardware upgrade from a Siemens Trio to Siemens TIM Trio that occurred during the span of the data collection periods for these different studies. We had multimodal data, and the studies differed on the modalities collected, the acquisition sequences, and the scanner software and hardware. Standard data processing was done with optimized modality-specific QA and pre-processing pipelines to compute modulated gray matter concentration maps, cortical thickness and peel surface area maps, FAMD and tensor mode maps, and multi-source interference task brain activation maps. These were all fed into linked ICA, and more details of how we actually implemented this and achieved a decomposition that separated out signals from noise are provided in our paper. The performance of our technique was assessed by artificially constructing 2 groups of healthy controls based on scanner. In other words, healthy controls pre-TIM upgrade and healthy controls post-TIM upgrade. If you do a 2-group t-test to look at group differences between those 2 sets of healthy controls, there should be no group differences because they're just healthy controls. So any group differences would be due to the TIM upgrade, for example. We tested linked ICA denoising with 3 different regression methods to denoise the data, including hard regression to remove the full variance associated with the subject loadings on each noise pattern, hard regression removing the full variance associated with the spatial maps for each noise pattern, and then regression to remove the unique variance associated with the noise components. And we compared our linked ICA denoising method with individual modality ICA denoising and GLM regression with covariates encoding study and scanner effects. So here are some results. We found 3 noise-related components, which were multimodal spatial patterns, or in this case, primarily dominated by gray matter concentration, but the loadings for these patterns were associated with scanner software version and with study effects. We denoised these effects from the data using the 3 different regression methods. And with ICA-based denoising, the effects were also reduced. The technique that provides the best performance in mitigating most of the effects is linked ICA denoising with hard regression using the subject loadings. And so in these 2 regions, we can see the peak z-values, or the average z-values within the voxels in that region being quite high with no denoising, and they trend down to 0 with our linked ICA denoising. We also showed in the paper that we can retain effects of interest. So, we were interested in cannabis, heavy chronic cannabis use, and this just shows a cerebellar component that we saw prior to denoising that was associated with chronic cannabis use that was still there after denoising, and that was also replicated using the Human Connectome Project data set. So we can implement this denoising and retain effects of interest, and even enhance our ability to detect other effects of interest that were missed when all of that study and scanner variability was there in the data. And then Dr. Lee also has a work in progress denoising side effects from the ABIDE 2 autism data set. So I'm just going to show you some very preliminary results here. And like I said, this is a work in progress where she's denoising these data and then using them to study autism. And so she did data fusion of different modalities for fMRI, namely amplitude of low frequency fluctuations, fractional amplitude of low frequency fluctuations, gray matter concentration, cortical thickness, and peel surface area. She did a decomposition at several model orders and then picked one where the scanner, where the side effects were best separated from the variables related to autism. 3 site-related components emerged from the analysis. I'm just showing one here, and this is the multimodal spatial pattern representing some global effects in the fractional amplitude of low frequency fluctuations and gray matter concentration and cortical thickness that were really driven by data from one of the sites included in the study. And so I'm not sure exactly what's driving this, what's different about this site, but this is just one of the patterns. So in conclusion, linked ICA with hard regression using the subject loadings performed well across all modalities and brain regions and generally outperformed GLM and single modality ICA. And this may be for a variety of reasons, but a couple are that scanner-related effects may be better captured by linked ICA and better removed using multivariate regression. In contrast to ICA, which has to be done on every modality separately, linked ICA is done only once. So that's definitely an advantage. Even though it's somewhat challenging to implement, you only have to do it one time. But further testing and optimization is needed, namely to look at different criteria for identifying noise-related components, looking at different modalities, and then how to implement this technique using super big data where you have a large number of variables of interest and a large number of confounders. Our approach has great promise for overcoming limitations of existing methods for addressing scanner variability and may have great value for combining data from recent large multi-site neuroimaging studies like the HCP studies, the UK Biobank, ADNI, and the ABCD study, which is a longitudinal study collecting data for more than 10 years in 10,000 kids at multiple sites across the US. Last, I would like to thank Wanji Lee, who is really pivotal in getting this method to come to life, and for my collaborators at McLean Hospital and University of Arizona for donating their cannabis-related data sets, and my collaborators in Oxford, and particularly Adrian Groves, who wasn't involved with this study per se, but worked with me a lot to teach me how to use the linked ICA and how to implement that. And then last, I would like to thank the NIH.
Hi, I'm John Murray, an assistant professor at Yale University, and I'm very thankful to be able to present to you today, along with Marcus Helmer, our talk on stability of CCA and PLS for cross-modality multivariate associations in population neuroimaging. My disclosure is consultation to Blackthorn Therapeutics. And I'd like to start with acknowledging the team who did this work, primarily Marcus Helmer, an associate research scientist in my lab, and he will explain the results in a few minutes. So here are our collaborators and our funding sources. I'd also like to point out that we have recently released a preprint and software package related to this talk, and therefore, if you'd like to learn more, I would encourage you to check out our bioRxiv preprint, as well as our software package called GIMR for calculating CCA and PLS stability. Okay, so let's start with a review of what canonical correlation analysis, or CCA, and partial least squares PLS are. So the key assumption here is that our data set is comprised of two feature spaces of interest, each of which is multidimensional. In the case of population neuroimaging data sets, our data set X feature space may be the set of neuroimaging features, and our data set Y feature space may be the set of behavioral or demographic measures. And each subject, each sample, lies within both of these feature spaces, right? For each subject, we have both neural and behavioral measures. And the goal of methods like CCA and PLS are to discover what are the relationships between these two that explain individual variation, right? So in this case, we would like to define weights as an axis within each feature space, such that when we project subjects along this axis to derive scores, in some sense, we are maximizing the association between these two feature spaces, right? We're maximizing, say, brain and behavior association, and then we can go and inspect and learn what the properties of those weight matrices, weight vectors are. So the key distinction between CCA and PLS is that CCA is maximizing the correlation between these sets X and Y, whereas partial least squares, PLS, is maximizing the covariance. And therefore, these are very related methods, although they would give rise to distinct weight vectors and scores due to the different objective that they are maximizing. So these two approaches of CCA and PLS have been applied to discover brain behavior associations recently in large population neuroimaging data sets. And the paper that really kicked off this trend is by Smith et al. 2015 in Nature Neuroscience, that was examining data as part of the Human Connectome project. And here they were analyzing the relationship between resting state functional connectivity measures and behavioral measures. And so typically the way that one assesses results and the statistical significance of a CCA analysis is by permutation of the sample labels. In this case, the subject IDs between neural and behavioral feature spaces. Rerunning CCA, you therefore get a null distribution, and you can see how many of your CCA modes, for example, live beyond this null distribution. So in the case of Smith et al., they found 1. And the key goal of these kind of CCA and PLS approaches to discover brain behavior associations is really to estimate and interpret the weights or loadings that mediate these associations between feature spaces, right? And so in the example of this paper, they wanted to discover what are the patterns of resting state functional connectivity edges here that mediate the relationship to the sets of behavioral measures. And therefore, by seeing, which features in the neural data set relate to which features in the behavioral data set, we're able to then discover the nature of that brain behavior relationship. As I mentioned, there's been growing popularity in these approaches, not only to study healthy human variation, but also application to neuropsychiatric populations. When we look at the literature of applying CCA to brain behavior relationships, we see a real explosion over the last 5 years. And I think that this literature raises important statistical questions related to the suitability of CCA and PLS, and to what degree we can interpret these results. And that's because most data sets actually do not live in the regime of large data set size, right? Most do not have a large number of samples for the number of features, typically 1 to 50. And therefore, that can lead to overfitting in this high dimensional multivariate data analysis. And we see signatures of this overfitting, such as high train set correlation, but low test set correlation, and permuted null correlations, which are near the train set value. And so the question that we addressed in this study is, when are CCA and PLS in this overfitting regime, and what are the consequences of that? And so the approach that we took is one of generative statistical modeling of multivariate associations. And so we wanted to develop a mathematical framework which could simulate synthetic data with known constrained properties. And then we could study to what degree the data analytic methods of CCA and PLS are able to recover the ground truth properties of our synthetic data. And so in this case, what we have to define for our generative model is the joint covariance matrix of these feature spaces, right? Not only the variance within, say, neural and behavioral feature spaces, but the between set covariance as well. And so we did that in 2 steps. First, we constrained the variance spectrum within each feature space, within, say, neural and behavioral, by empirical data. And so in this case, what we found is that the variance fall off is well fit by a power law, and therefore we can use the exponent of that decay to determine our within set covariance determined by actual data. Now for the between set covariance, here we made that such that we can constrain that with known ground truth. In other words, we can set this covariance such that the sample data have CCA or PLS solutions with specified association strengths and weights that we can control. And therefore, we know the ground truth of, in the limit of infinite data, how strong is the association between brain and behavior data, and what weights, what features mediate those relationships. And therefore, with our synthetic model, now we can generate synthetic data and see to what degree we're able to capture the ground truth properties that we specified. So now I'm going to let Marcus Helmer explain what we found when we applied CCA and PLS to our generative model.
We used the generative model to create synthetic data sets that perform CCA or PLS on these data sets and compare the estimations with the true results. And to do that, we used a variety of different metrics. We used the statistical power to detect a non-zero association strength. We looked at the association strength itself, and we also calculated a weight error, a score error, and a loading error. And across all these metrics, we immediately see that sample size had a strong impact. To unpack that a bit, let's first look at statistical power. Here we see that sample sizes that are sufficient to achieve high power are not necessarily sufficient to also achieve low errors for the other metrics. Second, let's look at the association strength. And here we see that for small sample sizes, the association strength was consistently overestimated. And interestingly, the estimated values were also pretty much indifferent to the true underlying value of the association strength. Third, let's look at the weight error. And similar to the score and loading error, the weight error decreased strongly with sample size. To get a better feeling of how variable weight vectors were, we used a generative model to create multiple data sets that should have the same weight vectors. When the weight vectors were estimated, however, with a CCA or PLS, we found that the estimated weights varied quite a bit, as shown here in the confidence intervals. We quantified this similarity with the average pairwise similarity of the weight vectors, which we denoted as weight stability. For CCA, weight stability was quite low for small sample sizes, but then increased towards 1 for larger sample sizes. For PLS, however, weight stability could already be quite large, even for small sample sizes. And that was despite the fact that weight errors were also large. So how did it come about that PLS weights seem to be inaccurate but stable? And to answer that question, we calculated the overlap of weight vectors with principal component axis. And we noticed that PLS weights tended to have a strong overlap with the first principal component axis, and that this effect was especially strong for small sample sizes. And we did not observe this PC1, this principal component bias for CCA. Finally, we also found similar effects across a variety of different parameter scenarios. Next, we asked whether these results that we found for synthetic data would also hold in empirical data. And for that, we turned to brain behavior relationships and neuroimaging data. We used data from the Human Connecting Project with about 1,000 subjects, as well as data from UK Biobank, where we had 20,000 subjects. And we related neural features to behavioral and demographic features. All data sets were deconfounded, and then principal component analysis was performed to reduce each data set to 100 principal components. And these reduced data sets were then entered into a CCA or PLS. Let's first look at the results for the HCP fMRI dataset. And to investigate the stability of the CCA and PLS, we repeatedly subsample the data and then calculate a number of metrics as a function of the subsample size. The metrics that we looked at were similar to the ones that we also used for the synthetic data. So here we are showing the in-sample association strength, and we're also showing the cross-validated association strength. We're also showing the weight stability and the weight PC1 bias. And across all of these metrics, the results appear pretty similar to the results seen in synthetic data. The second data set we looked at was HCP diffusion MRI data. And again, all these look very similar. And the third data set we looked at was UK Biobank fMRI data. And once more, all these look pretty similar. One thing to point out, though, is that for UK Biobank, we had 20,000 subjects in contrast to just 1,000 for HCP. And here we see that the in-sample and the cross-validated canonical association strength have converged. Going beyond just these three data sets, we also looked at brain behavior, CCAs, in the literature more broadly. And we found 100 CCAs from 31 papers. For all of these, we extracted the number of samples per features used, as well as the observed correlation. And then we plotted these against each other, and we observed this very strong anti-correlation. And this anti-correlation indicates overfitting. Finally, we created a sample size calculator for CCA and PLS. And to do so, we created a zoo of synthetic data sets where we vary the sample size, the number of features, the true correlations, and the within set variances. And for each of these cases, we then determine the sample size such that a predetermined level of error would be achieved. And as all of these calculations are computationally quite expensive, we also developed a linear model that would allow us to quickly predict sample size even for new parameter values. And to conclude, we developed a generative statistical framework for simulating multivariate brain behavior associations. We found that CCA and PLS are often prone to overfitting. We saw that a statistically significant detection of a CCA or PLS association does not necessarily imply a reliable estimation of the weights. We also saw that weight stability in PLS can be misleading due to the bias toward the first principal components. We also saw indications of overfitting in the brain behavior CCA literature, and we created sample size calculator for CCA and PLS. And with that, I'd like to thank you for listening, and I'd also like to thank Lisa Nickerson for organizing the symposium. Thank you.
Hello, everyone. I'm very glad to have the chance to give a talk in this multimodal fusion symposium. My name is Wei Kang Gong, and I'm a PhD student with Stephen Smith at Oxford and Christine Beckmann at the Donder Institute in Nijmegen. My work concentrates on creating tools and techniques for analyzing large-scale population studies. Today, my topic is Big Data Fusion for Phenotype Discovery in Large-Scale Population Neuroscience Studies. The neuroimaging field is facing a big data era. Imaging neuroscience has seen a transformation in recent years. While initial image studies involved maybe 30 to 100 subjects, with the projects like the Human Connectome project, the field has moved forward. Currently, as part of the UK Biobank image extension projects, some 100,000 subjects are being imaged. And while such projects offer tremendous opportunity to better understand the brain in terms of linking brain structure and function to clinical, cognitive, and demographic variables, However, computational feasibility remains a challenge for such big data sets. Currently, the most widely used approach for compressing such image resources is to use image-derived phenotypes, typically scalar quantities that summarize imaging features into simple numbers. An example would be the mean cortical volume derived from a subject's anatomical image, or the mean Z statistics under task within a certain cortical area derived from task fMRI. The IDP have been shown very good heritability in our recent paper. This knowledge-based approach is simple and efficient, and effectively reduces the high-dimensional data into interpretable, compact, and convenient features. However, there may be a large loss of information due to IDPs not capturing important source of subject variability, as well as ignoring cross-modality relationships. And therefore, an open question remains if we can do better defining these IDPs in a data-driven fashion. Further, such large-scale image studies deliver multiple types of data, and from this, we can derive an even higher number of phenotype maps. For instance, a single diffusion MRI experiments give rise to multiple maps of scalar values, such as FA and MD. Classically, these derived maps would all be analyzed separately. Our approach for simultaneously integrating and analyzing such multiple modalities was to focus on common modes of variation across the population. Given these multimodal maps, together with subjects' demographic, behavioral, clinical informations, known as non-imaging derived phenotypes, we aim to discover a low-dimensional representation of the brain maps. Each of them represents a specific set of brain areas covariates together related to IDP and IDPs. In 2010, our group introduced linked ICA. In a nutshell, that method decomposes given data modality through independent component analysis. Flickr does separate ICA in each modality, but constrains that subject cores are shared across modalities. Therefore, for each component, there is one common subject cores with modality-specific spatial maps. In addition to this, Flickr also estimates a positive weight per component per modality, representing the contribution of a component on a given modality. Flickr achieves this by the variational Bayesian algorithm. The spatial independence constraint is achieved by a mixture model prior. For UK-Baumann data in this work, we analyzed 14,000 subjects across 47 modalities, each being represented across over 150,000 work cells. Jointly, this is 800 gigabytes of raw data. If we were to use vanilla Flickr on this data, we would require approximately 1 gigabyte of peak memory and 1.6 thousand hours of computation. That is larger than 2 months of non-stop computing, even for this subset of UK-Baumann subjects. So in order to address this, we have modified Flickr and introduced 2 types of innovations. First, we introduce data compression in subject space. By this, I mean we analyze the subject-by-subject covariance matrix of the data and create what can be best described as eigensubjects. This reduces the dimensionality of the matrices fed into Flickr by a controllable amount. For those interested in details, I'm pointing you to this relevant paper here. Secondly, we combine this with compression in the spatial domain. Instead of using fixed atlas, we used what is called dictionary learning, an approach that allows us to define the data dictionary separately per modality. Again, I won't have the time for the technical details and only refer to your very relevant paper here. We have extensively evaluated those 2 approaches using simulated data across different signal-to-noise ratios. different level of compression, and different number of components. The big flicker with MIG-P processing give very similar results to the original flicker, and both flicker and big flicker accurately find the underlying ground truth in most cases. On the left, the highest signal-to-noise ratio case, a relatively low-dimensional MIG-P can achieve an accurate estimation of the component, while on the right, the high signal-to-noise ratio case, a high-dimensional MIG-P is needed. For dictionary learning, we tested the influence of signal-to-noise ratio, component number, dictionary dimensions, and dictionary sparsity. Figure on the bottom shows that in almost all circumstances, the optimal performance given by dictionary learning matches that of the non-reduced analysis. In summary, for the simulation, we can obtain very significant data reduction in both subject space and in spatial space through these two approaches. In real data, as there is no ground truth available, we tested whether the modes of big flicker have a similar prediction accuracy of NIDPs compared with the original flicker using 1,000 subjects from the UK Biobank. Here are some blind Altman plots that describes the differences in prediction correlations with the NIDPs in UK Biobank. In almost all cases, we see a delta R that is small but larger than 0, which means that our prediction accuracy is equal or even better when compressing the data relative to feeding into the non-compressed data to the flicker. We assume that this is due to some inherent denoising when compressing the data. For computation, on the 14,000 subjects we analyzed so far, depending on the number of components to extract, vanilla flicker required up to 1.6 thousand hours of computation and up to 1 GB of memory. While through our compression approach, we can fix the peak memory to about 50 GB and reduce the computation time to less than 100 hours. In the full UK Biobank sample, we estimated that without compression, we need some 6 GB of peak memory and 12,000 hours of computation if we use vanilla flicker. While with big flicker, we can obtain a decomposition in 1% of the time and less than 1% of the memory. Ultimately, this means that instead of using 1.5 years and a supercomputer for this analysis, we will be able to analyze 100,000 subjects within 5 days on a laptop. In terms of the prediction, UK Biobank contains over 8,000 non-imaging derived phenotypes. The plots shown earlier related to an analysis of all of these NIDPs, but I will give you a few examples. For example, big flicker has a significant improvement on prediction accuracy on fluid intelligence, whereas the R of big flicker is 0.26 and R of the IDP is 0.23. And for age started wearing glasses, the R of big flicker is 0.15, while the R of IDP is 0.12. And for handiness, the R of big flicker is 0.23, as the R of IDP is 0.12. And finally, for the hypertension, the R of big flicker is 0.22, while the R of IDP is 0.18. At the same time, all of these correlations can be mapped back into the brain so that we can investigate which data modality gave rise to good predictions. For instance, when it comes to handedness, the lateralized modes are involved, including resting state language networks, task activations, surface areas, and wet matter tracks. Well, if you are interested in age starting wearing glasses, we can predict this from both task and the rest data. Several resting state connectivity and task modalities showing associations in primary visual areas, which is consistent with the fact that this is a vision-related health variable. So the approach introduced so far works in two stages. First, we use the optimized Flickr approach to estimate subject loadings. And then the second stage, we link these subject modes to the NIDPs. A natural question arises, can we do better? Yes, we can. Instead of separating the decomposition and prediction stage, we can link these two via a joint cost function. Not only the data reconstruction, but also the prediction part now involved in the estimation. And the cost function now can involve more than a single NIDPs in the estimation. And that is, we can use multitask learning so that we can steer the decomposition towards features that generically have high predictive power. Again, we have evaluated this over 16,000 NIDPs and in 40,000 subjects in the UK Biobank. Here is some examples. For example, we have looked into the prediction of IQ. Well, Big Flickr already does better than predicting IQ from handcrafted IDPs. We observe that this is further improved by adding supervision to the cost function, which is shown in the third bar. Further, by including some NIDPs that highly related to IQ, we can further improve the predicting of IQ by multitask learning, see the fourth and fifth columns. A similar trend has also been observed for the NIDP hypertension and for handedness and for age-starting wearing glasses. Note that these NIDPs are selected because Big Flickr has a very good performance. In summary, Big Flickr enhances the ability of original Flickr for multimodal fusion in the large datasets. The modes learned by Big Flickr have an increased prediction accuracy of NIDPs compared with traditional methods such as handcraft IDPs, ICA, the original Flickr. Adding supervision to Big Flickr further helps improve the multimodal learning. If you are interested in using our algorithm, please refer to this GitHub link. And if you are interested in knowing more detail, please refer to our paper. And that's all for my talk. Thank you very much for listening.